This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1007, recorded on May 12th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here, one of my apps says that it's 80 Fahrenheit, and the Norwegian weather app says that it's 23 Celsius. And both of them say that it's cloudy throughout the day. It might rain a little bit later, but it's a pretty good day, pretty warm.
Yeah, we have 30 Celsius here. It's like 86. It's really warm today, but I'm inside, and the air conditioning's kicking in now, which I don't need. And it's noisy, which I don't like. I can't control it. Oh, well. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 83 degrees, overcast, rain in the future, according to the weathermen. And they seem really pretty convinced of this this time. We could, we're like 30%, 15 to 30% behind in our annual rainfall, depending on who's counting. And we could use this, and this may go a long way to making it up. We're looking at maybe a couple of inches over the next couple of days, which would be great.
And from Montreal, Canada, Angela Mingarelli.
Hello, everyone. So it is 75 degrees Fahrenheit here, I checked, which is 24 Celsius. And it's actually surprisingly warm. We're very excited in Montreal because the temperature has not gone below 5 degrees in about a week. And the weather next week, the weather prediction looks good. It looks like we're going to stay around the teens during the day. So summer is finally coming. We're excited.
Well, cool. Spring, isn't it supposed to be spring first?
Well, true, but we kind of skip. We don't really have a lot of spring in Montreal. We go from like 0 to 25 in 3 days. So
I remember when I lived in Buffalo, a visitor getting off a plane sometime in early May and kind of looking around and telling me, it's spring everywhere else.
Yeah, that's right.
So, Angela, with you giving those temperatures, it really confirms what I thought, which is that the Norwegian weather app is not telling the right temperature. So it's 27 in Celsius here, 80 Fahrenheit.
Yeah, when you said 23, I was like,
it doesn't make sense.
I Googled it. Yeah, I was like, I don't think that's correct.
Yeah.
But the Norwegian weather app, we got to fix it.
Yeah. Yeah.
So, Kathy, last week after TWiV, I found, I didn't find last year's lanyard, but I found the 2 previous years. So I decided to bring Minneapolis lanyard, which is like,
it's the same format. It's the same size as the one we got. So the insert should fit just fine. And maybe the other one will still turn up, Maybe it's in one of your suitcases.
It could be, but these are all hanging on a doorknob behind where I record at home. And which is the one after Minneapolis?
The one last year is just plain black. It doesn't say a year or a place.
That was in
on
Wisconsin, right?
Yes.
Yeah, I don't have that one. I have Minneapolis, which was the year before, right?
Right.
And then the year before that was Maryland, correct?
Right.
I have both of those, yeah.
Maybe it's hanging on your door at Columbia, on the back of some door.
Well, then it's gone because I cleaned that out. I don't have a door anymore at Columbia.
Right.
Anyway, what else should we know about ASV?
Well, definitely try and find your black neck pouch or lanyard from last year. Also, I think I've already mentioned this, but there's going to be this documentary, Virulent, which is going to be shown during the dance. So if you don't want to dance, you can go watch this movie. And there's a 4-minute teaser that's available. And I put the Vimeo link in here for that. Maybe Vincent will put it in the show notes, but it'll definitely be in the mobile app and some of your meeting materials once you get there. So
it's the one time a year I get to dance. I'm going to go to the dance.
Yeah.
and it's the whole thing, the whole time. And you better get a good band.
Well,
or whatever it is.
Mark has been working on it. He sort of auditioned on tape and I think went in person at least 3. And I think he's down to the wire. And I think it's going to be an Athens homegrown band. But he hasn't signed the contract yet. So.
It's good. A band last time we had a DJ, right?
Yep.
So,
Yep.
we still have an opening for a research assistant with Amy Rosenfeld at FDA to work on enteroviruses, BSL-2 conditions, animal models, the role of cross-species immunity. You know what's interesting? Just a side thing. This is science. So last year, Amy published a paper saying that there's extensive cross-neutralization among different species, like different enteroviruses, poliovirus, Entero68, even rhinoviruses. She's finding all these papers from the 40s where they report the same thing. She showed me one today. And I said, maybe we should do this on TWiV. She said, well, it's from the 1940s. You might not want to do that. I said, why not? They do all kinds of cross-neutralization. The 3 serotypes of polio are not absolute. You can get cross-reactivity in neutralization assays in cell culture. They did it in mice, and they did it in non-human primates. So this is something that's been known for a long time.
I think doing old papers is cool. We
I was going to say too.
We did several of those. You know, we did Jenner's inquiry. We did the original phage paper. I think we did an original Rouse sarcoma virus paper or something like that. That's really fun.
Mm-hmm.
This is they're just tables and tables of neutralization assays, right?
Yeah,
that's what they had. They didn't have PCR, didn't have genome sequencing.
Right.
Really interesting. Anyway, so this position is to work on these kinds of cross-reactivities and figure out what they do. Coronaviruses like Entero68, poliovirus, human rhinoviruses. So I will put a link in the show notes to a PDF where you can find her email and a little more description if you're interested. All right. Now, I had an idea to keep this episode short so we could do some email, but it's not happening. I'm sorry, because I just found stuff. You just look and you get so much cool stuff. Number one, there's an interview in polioeradication.org. If you have never gone there, it's an interesting site where they keep you up to date with news about what's going on with the eradication. And this one is it's from March 30th, 2 years since rollout of novel oral polio vaccine type 2, NOPV2. How's it all working out? And it is an interview done by Simona Zapersky with Ananda Bandyapati, who is in the Gates Foundation. But this is an interview where he's going over what's happening. So remember, OPV2 is problematic. Oral polio vaccine, serotype 2. It's problematic in part because we declared it eradicated a number of years ago, and then we removed the type 2 component from trivalent oral polio vaccine, and now vaccine-derived type 2 polioviruses are circulating extensively. They've caused hundreds of cases of paralytic polio in Africa. So a couple of years ago, the Gates Foundation actually said, let's make an OPV that can't revert. All right, so they funded a couple of labs to do this, and we talked about it. We did the papers. It's called NOPV2. It's genetically modified in multiple ways. So one of the problems with reversion is the 5' non-coding region has an important determinant of attenuation. That means it has a mutation that prevents the virus from paralyzing you, okay? And that reverts very quickly when you drink the virus. And so they tried to engineer changes in the genome to prevent that. So they made a lot of changes around that 5' non-coding region. Then they modified the RNA polymerase in 2 ways to make it make fewer mutations and to make it recombine less. All right.
Who's they, Vincent?
A number of laboratories. Raul Andino was one. Andy McAdam in the UK, and some others. And this was made in the lab. It was tested in cells in culture. It was tested in transgenic mice for the polio receptor. It was then tested in people in the poliopolis that we talked about.
Yep.
And now it's been put into 600 million kids in Africa.
That's an amazing
number
over the course of like how long?
Like 2 years.
2 years.
2011, Endeavor was launched to develop. So I think it's 2 years because it took a long time for them to develop it. And not too long ago, we had 7 cases of paralytic vaccine-associated polio. In other words, this virus caused paralysis in 7 kids. Now, if you were still using OPV2, it would have been 40 or 50 kids. Okay, so it's an improvement, but it's not 0. And the reason I'm bringing this up is because he says, so no one's released the sequence of these viruses that these kids have shed. CDC has the sequence, but they're not releasing it. I don't know why. I went on Twitter this week, and a rare appearance on Twitter, and I said, CDC, please release the sequence. Scientists would like to look at it. More eyes are better than one eye, right? So he says here, these viruses that came out of these kids likely derived from double recombination events with human species C enteroviruses. All right, and that's all he says. So what does that mean? First of all, when you take OPV, it recombines with enteroviruses that we all have in our intestines. And what comes out after a few weeks are all recombinants with OPV. With enteroviruses. We know this. We've known this for years. So apparently these NOPV2s have recombined. Here's what I think happened. I think the polymerase is gone. The engineered polymerase is gone with the changes that make it have more fidelity and less recombination. I think those are gone. And I think the capsid is maintained because you're looking for Sabin 2, so that's still there. And they had moved an element to the 5' end to prevent recombination. I think that's gone too. This way he says there's a double recombination event. I think the original 5' end with the changes they introduced, I think that's not changed. So it's not really revertant. It's a recombinant now that has somehow acquired neurovirulence properties by virtue of these other sequences. So obviously it's really interesting to know the sequence, CDC. I mean, why can't you release it? What are you afraid of?
I wonder if you can get rid of the recombination potential by recoding the thing. Changing all the silent positions and stuff.
Thank you.
So that there's no homology or reduced homology that would promote recombination.
Good thing you reminded me. There were 2 original candidates for NOPV2. One was this one, and the second was a recoded one from the CDC, where they made thousands of base changes.
Right.
And so maybe that wouldn't do the recombination. I don't know. That's a good question.
It could be that what you need is, some combination of them. But I mean, the recoded guy ought to be, maybe it's already been done, because I would imagine that this would be one of the motivations, ought to be assessed for its recombogenicity. Whatever.
Recombinogenicity? I don't know. Anyway, I want CDC to release this sequence because I would like to know what it looks like. And other scientists would like to have a look at it and weigh in, right? Come on.
So, Vincent, I'm sorry. I can't hold all this stuff in my head. And if I can't, others
Listeners
can't
won't.
either.
Okay.
Type 1, 2, and 3. 2 was supposedly eradicated, right?
Yeah, 2 and 3 are eradicated. They're declared. We don't see any more paralytic polio. But look, sewage surveillance is almost non-existent, right? So I think you have no disease caused by types 2 and 3. Whether the virus is gone, not so sure.
And 1 is still out there?
1 is still circulating, causing disease in Pakistan, Afghanistan. It's just spread to Malawi recently.
And so what is typically in vaccines now that are distributed, in particular the oral vaccines?
So the oral vaccine is bivalent, types 1 and 3 only.
Okay.
And when you have an outbreak of type 2, they go in with monovalent type 2, NOPV2 in this case, and they try and mop it up. In the U.S., of course, we use trivalent IPV. Many countries, U.S., many countries in Europe, Israel, are using trivalent IPV.
And
I think what we have to do is think outside the replicating poliovirus box for vaccines. And if there's problems with the inactivated vaccine because of cold chain issues, then let's figure out a different way. Let's use a different virus
I agree.
platform if you want live virus to get you cell and humoral immunity, or do some RNA vaccine, or do some subunit vaccine. But
I agree.
trying to outsmart the virus,
Not
the
happening.
polio
Right?
virus, is not going to happen.
I totally
Especially
agree.
with all those other enteroviruses around. I mean, I wrote that, you put this article in here for us to read, and I wrote that at the end. It's like, why are we still focusing on these attenuated viruses?
Do you know if there are any other efforts, Vincent, for other platforms for polio virus?
I've heard
Vaccination?
people talk about it, right? Gates is totally invested in this. If you read this whole article, this guy basically said, it's fine, it's all okay. It's going to be fine, we're going to wipe it out. And it's just wrong. This is not working.
Yeah, I mean, the hypothesis is that if you can get the vaccination done right, where you get everybody vaccinated and everybody vaccinated again and everybody vaccinated again right away, and you get 100%, 150% coverage,
Yeah.
then it'll work.
But it's not
how
feasible.
realistic is that? Yeah.
Yeah.
I mean,
it's so frustrating.
Personally, I think we should all be using IPV for now until we develop something else, because, OPV has been delicensed in the U.S. We switched from OPV to IPV in 2000, and it's delicensed. Why? Because it causes polio. So if it's not good enough for us, why is it okay to give it to African kids and get them paralyzed? I don't agree with that any longer. I don't think that's right.
Isn't it because I think we spoke about this on another episode, because having at least some sort of vaccination is better than not having anything at all, right? If they can't have IPV, it's better to have OPV and have, instead of 40 cases, 7, which is still...
the more I think about it, though, I mean, you're giving kids something that's how they justify it. Oh, there's fewer cases of vaccine-associated polio, and it's preventing a lot of wild polio. But the kids take the vaccine. Can you imagine you're their parent and they get paralyzed from it? I just don't think it's a good idea. And you could do IPV globally. I'm sure you could figure it out.
IPV being effective suggests to me that all you really need as an antigen is the capsid antigen, right?
That's right.
Is it known whether you? I mean, there's more than one capsid protein. Is that correct?
Yeah, there are 4.
There are 4, but there's one major one, right?
You need all 4 to make a capsid.
You need all 4 to make a capsid, but to immunize. I'm thinking of a protein vaccine, an mRNA vaccine, right? I'm wondering what you would need.
You could design an mRNA vaccine to make the capsid. Okay.
Right.
So you'd have to have all 4 proteins, and the thing is, they're processed. So
you'd actually need the capsid.
Yeah, you need the capsid.
So it would have to be a virus-like particle.
You need a virus-like particle, totally.
Okay.
Yeah. So you have to figure, you could do that with an mRNA vaccine.
Okay.
There are other ways. You could use adenovirus vectors, whatever you...
You could do however they did it for HPV, right?
Yeah, for HPV, there was one major capsid protein, and making that alone gave you capsids. So
right.
It's not the case with polio. If you just produce VP1, you're not going to make empty capsids. But there are other strategies for making them that we've talked about, and that could be done. But the WHO and CDC and Gates are all still bullish on this OPV doing it. And back when they started making an OPV2, okay, they thought if they did these modifications, it would help. And it certainly has helped a bit. But okay, give up now. Do something else. I just personally don't feel we should be paralyzing kids with a vaccine. All right. If it's not good enough for us, it shouldn't be good enough for anyone. But that's just my personal feeling. But I want the sequence. I want to look at it, right? And see what happened, what kind of recombination occurred. Isn't that amazing? You change the polymerase. And, I'm thinking back, why didn't they think of this? It's because every OPV that comes out of kids is a recombinant.
It's not nice to fool Mother Nature.
Do you think that's why they don't want to release the sequence? Because in the eyes of at least, I don't want to say the lay public, but certain people, it could be seen as a failure and something that they should have known. And then if they release a sequence, they see that it's this recombinative event could make them seem that they should have foreseen this, I guess. And they don't want to show that. I don't know. But
I mean, I don't blame them for not foreseeing it. But now that it's done, I don't think you should try and push it. I mean, they're just continuing to immunize, and they're saying, "Oh, we'll take care of it. We'll stop the circulation." But he says these numbers are based on an ongoing assessment and may change as we continue monitoring. The other thing is very little wastewater monitoring, so you don't even know what's there. You know, if they say, "Oh, it's these strains are limited," I don't believe it. I don't believe it. Anyway, so it's really an interesting situation. I didn't think that it would come to this, frankly. IPV, everybody IPV, or microneedle patch. Apparently that's not as good, because that's too bad, because microneedle patch would be cool, right? Put it on with a Band-Aid.
And that is the thing with IPV, is that it has a cold chain issue.
It has a cold chain issue, and it's got to be injected, yeah. But we did do massive immunization with COVID vaccines injected, right? So it's not impossible. Although Africa didn't get a lot of COVID vaccines.
That's true.
Right?
So this is a little in the weeds here, but I forget. Man, I've been retired for a long time. You make a polyprotein, right? That's got coding sequences for all 4 separate antigens, and that gets processed, proteolyzed, right?
And it folds up, yeah.
The protease, is that, so if you made an mRNA vaccine, would you have to, is it autocatalytic? Is there a protease associated with that? No,
it's a protease, and you don't want to put that in because that's toxic, okay? The protease is going to do things that you don't want.
So the mRNA vaccine, would you have to do a quadrivalent mRNA vaccine and do the individual open reading frames?
There's a 4 amino acid sequence that does start-stop. Stop-start. So NPGP. If you put NPGP between the 2 coding regions, the ribosome will stop, release the first polypeptide, and then start the second one.
Cool.
So you
can like that.
Isn't that cool? So you make one thing with the 3 start-stops, and you make the 4 polypeptides. Yeah, that's how to do it.
Where'd that start-stop thing come from?
Another picornavirus.
Okay.
So, from the cardioviruses, it's between the capsid region and the rest of the genome. There's no protease there. Normally, there's a protease there that cleaves between the capsid and the rest of the polyprotein. Okay. There's a sequence, NPGP.
Crazy.
Asparagine, proline, glycine. Wait, NPG, glycine, NPGP, proline. And you'd put that in between any 2 proteins, and the ribosome stops, releases one, and it keeps going. Isn't it? That's just
glycine and proline. It's like putting a great big knot in there. Okay, and the ribosome goes, to hell with this. So, is it known that if you make those 4, well, I guess it does in this other picornavirus. If you make those 4 independently like that, it will self-assemble?
Yeah, it will. Sure.
Okay. Well, get on it. That's the answer.
Yeah.
So there's just one more thing, all right, that the empty capsid is actually antigenically a little different from the capsid with the RNA in it.
Ooh.
Okay? So empty polio doesn't give you the same neutralization as polio with RNA in it. But Andy McAdam figured out what amino acids to change in the empty to make it look like the RNA containing. So that's ready to be done. It can be done. But here's what we're up against. WHO wants everybody to destroy their stocks of polio. How can we do this stuff, right? So just stop the destruction stuff, because more research needs to be done. And anyway, the sewers are full of polio. So what are you worried about?
Yeah,
so what if a lab has it?
Reminds me of the let's get rid of the remnants of smallpox debate.
Yeah. I haven't heard that in a while, right?
No.
Okay. All right. So the next thing, the other night on live stream. By the way, we had Dan Wilson on my live stream this week.
Oh, cool.
That was fun.
Yeah, he's good fun.
And someone said, "Hey, there's a raccoon distemper outbreak in Canada. Can you talk about it?" So I said, "Oh, Angela's going to be here. So what's up? What is raccoon distemper?"
Yeah, so raccoon. Well, it's not specifically raccoon distemper. It's actually canine distemper virus, which a lot of people, those that have dogs, know that this is a virus that dogs are actually vaccinated against when they're puppies, around like 2 months old, and it's normally part of this this vaccination called the DAPP, so the DAPV, which is multiple viruses, including the canine distemper virus. And to give you some etiology about the virus itself, it's a single-stranded RNA virus, and it's actually a paramyxovirus. So its human counterpart would be, closest would be measles. And then rinderpest, which I know we spoke about recently on TWiV as well, it's another paramyxovirus in cattle. And basically, and dogs, like I said, are vaccinated against this, but raccoons, so it can infect many different mammals, actually. So it can infect many animals of the order Carnivora. So foxes, skunks, ferrets, even seals, because a lot of people don't know this, but pinnipeds, seals, are actually part of order Carnivora. And there's actually a phocine distemper virus, which is the most closely related to the canine distemper virus. And it has tropism. Phocine, like for seals. Yeah.
Okay.
PHO.
PHO, sorry. Yes. PHO. And it has, tropism throughout the body and multiple tissues, the main ones being the gastrointestinal tract, the respiratory tract, and the central nervous system. So in Toronto, unfortunately, here in Canada, there are a lot of raccoons. There are hundreds of thousands of raccoons downtown. And right now there's an outbreak in these raccoons. And unfortunately, there's not a vaccine readily available for wildlife like there are with other vaccination in wildlife, like for example the oral rabies vaccine. There's also one being developed for Lyme's disease, so Borrelia burgdorferi, that's transmitted by deer mice, so the ticks that live on the deer ticks, that's also being developed. And yeah, so you'll see that these animals can apparently look like they have rabies because in the more advanced phases, they'll have neurodegeneration, they'll have ataxia, they'll be stumbling, they can have seizures. You'll see their eyes are kind of, there's a yellow discharge from their eyes. It's very sad. It's tragic. And in dogs, some of them can survive if they don't, if it's not lethal. But a lot of the time it is lethal in mammalian species. And yeah, so right now, unfortunately, this is happening in Toronto. But I looked online to see if there are any efforts to develop a distemper vaccine in wildlife, but I didn't see any because they're very sporadic outbreaks. It's not like rabies that's maintained as like the reservoir species of the Lyssa virus. So yeah, hopefully. I mean, raccoons, even though some people call them a nuisance, we don't want any animals to die unnecessarily, and it's a horrible death. So for now, they're just culling all of the animals that have any sort of symptoms to try to stop the spread, because the spread is via air droplets. So any sort of sneezing, discharge from the animals, they transmit it very quickly from one to another.
So what's the case fatality percentage?
I don't have that in raccoons specifically, but it's quite high. Also, obviously, it depends on the age, like if the animal is immunocompromised or not. But yes, it can be very high. And in dogs, it's very high. And in, like I said, it also can infect pandas, other bears. Interestingly, I was Googling other species that I wasn't aware of that could infect. But yeah, hopefully it'll get under control quickly. Toronto
I have two questions. I saw something somewhere. I don't think I was able to see it when I clicked through, but it implied that panleukopenia virus in cats is also one of these. Is that a distemper or?
It's similar. Yeah, panleukopenia virus is a similar virus in cats. Although there is like a distemper form. So some felines can be infected with canine distemper virus as well, actually. So felines and large felines as well. Panleukopenia is specific to cats, but canine distemper virus, because we call it canine distemper virus, but as I said, it can infect multiple species in mammalian species.
What does panleukopenia mean? Is all your white cells are gone
all of your,
down?
Yeah, it affects mainly the white blood cells. Exactly. So,
and is the virus actually infecting hematopoietic cells, and that's the cause of that, or some precursor?
I'm trying to remember if there are inclusion, if it's actually in this. I don't remember if it infects specifically the immune cells. I think it does. I think it has to. I think it's actually it affects hematopoiesis.
Okay.
Let's see.
Kathy, I'm sorry I interrupted you.
Well, that's okay. My other question was going to be if there's these thousands and thousands of raccoons and they're a pest, are there any decent pest control measures, or is it like
it's
rats
culling.
in New York? And yeah, okay.
Yeah, you have to cull the animals. Yeah, they're euthanized because it's also, you want to stop the spread, right? And you need to stop the spread as quickly as possible, because then it can also infect other wildlife populations outside of Toronto.
Right.
It can,
right. But I mean, just in an ordinary situation when we don't have the canine distemper around, raccoons are still a problem, but there aren't any, like, you can't trap them, or I mean, it's just out of control, sounds like.
you mean just raccoons in general in Toronto, or in a big city?
In Toronto, yeah.
Normally, so you're not allowed to just go and kill a raccoon. So they can be trapped if, I know you can call like pest control, etc. If they're actively trying to enter into your house or like endangering your family, etc. But if you were to go and just kill one on the street, you could be fined, because obviously they're still living beings. Like, even though we call them pests, they're just trying to live. They're just animals, they're encroaching on our habitat. We've encroached on theirs. So it's,
so who's the pest, right?
Exactly.
That's so how come there are so many raccoons?
Good question. I mean, there's so much. I want to say there's a lot of green space as well, although they're not living in the green space. These raccoons are living in the alleys, in the garbage cans, in people's attics, actually. Because a lot of homes here have, well, of course, and in the United States, I mean, there's a lot of attics, right? So raccoons will make their way into someone's attic. And a lot of the time, the family doesn't even know they're living in there until they either go up one day after years, because they don't have regular access, right? I know one of my friends had a family of like 8 raccoons living in her roof for years, and they had no idea.
Cool.
So this is an anthropogenic thing, because if we didn't have cities, the raccoons wouldn't be there all cramped together, and they wouldn't be spreading this they'd be out in the countryside, far apart, right? It's our fault, as usual.
So when I looked over some of the information that you sent, I had particular eye for whether or not you could make a wildlife vaccine. And it looks like, I mean, there are some virus vectored vaccines, right? There's a canary pox vaccine, I think, and an adenovirus vectored vaccine. And I presume that these are single antigen vaccines, some sort of spike equivalent protein. Do you know? So it seems to me everything you need is right there, just to stick it in a bait and distribute it.
No, yes. But the thing is that, like, obviously cost is a major part of this, and having, is it worth developing a vaccine for something that is sporadic and something that's not going to necessarily affect because obviously if it's affecting domestic species, we already have a vaccine for domestic species, but animals like raccoons, trying to justify the development of a vaccine for raccoons or other wildlife that are known as pests, a lot of the time is much more difficult.
So under the circumstances, the vaccine would just be saving them to be culled.
Yes, basically. Which a lot of people unfortunately want anyway,
right?
Because they consider them like pests.
So listeners are wondering, does canine distemper virus infect human cells?
No, it does not.
Are the receptors known? Why doesn't it infect human cells? Is it a receptor thing or an intracellular thing, or I
don't have that information specifically, no. Okay. But I know that I specifically looked to see if there was ever any sort of human infection, and there's no human infection. I'm not sure. I mean, if it seems to only affect mammals in the order Carnivora, and outside of that, I mean, could it eventually infect humans if we put it? I mean, measles is obviously, it's a completely different virus, but it's the closest counterpart. Okay, that's like asking, why can't measles infect dogs? I guess.
I smell a gain of function experiment.
Exactly, there should be some sort of. You could pass it through some cells, see if, yeah.
It may be known. I know Paul Dupre works on distemper viruses, so he probably would know. But he's not here. Okay, thank you, Angela.
No problem.
Yeah, cool.
One quick note here. The NIH has reinstated the grant for coronavirus research of EcoHealth Alliance. Some of you may remember that early on in the pandemic, shortly after the president of the U.S. said the virus came from a lab in China, they terminated the NIH grant of EcoHealth Alliance, which was in part to collaborate with the Wuhan Institute of Virology and see if coronavirus is circulating in bats or had any pandemic potential, or at least it's some potential to infect human cells. And the funny thing is, I interviewed Peter Daszak in December 2019 for TWiV at a meeting in Singapore, and he explained what they were doing. So, you take the spike and you put it into this virus and you put it in cells. And Fox News took that clip and played it and said, this is the man explaining how he made SARS-CoV-2. Anyway.
It's insane
it's
that's out of context. No, of course.
Totally.
And those are things that are done in the lab all the time. It's necessary experiments that need to be done to be able to see if said virus can infect human cells. That's just...
So Trump said, take away the grant, and Fauci wouldn't do it. And then they got the head of the NIH. What was his name? Tom something? Tim something?
No. It wasn't Varmus at the time. It was
Collins?
Francis Collins.
Francis Collins. He did it. And this is totally wrong. You don't set a precedent for taking a grant away that you've got by peer review and all that. Anyway, they gave it back to them now. But so many conditions. They can't do anything in China. They can't do anything with Wuhan Institute of Virology. They can't collect samples from bats over there. And apparently their accounting is going to be scrutinized because there were some issues with that. But
so the way they said, sorry.
Not being able to collect new samples. What?
Yeah, that's what I was going to say. But is that only in China, or is that from any bat? Because it's not clear.
I think it's just China.
Yeah, it's written a little bit ambiguously in this Nature article, but that was my question also. I think it's only in China that they can't do it.
I mean, that's where the threat is, right? These the two outbreaks. Oh, well. So they should never have taken it away to begin with. This is a politically motivated thing. And that's what they say at the end. Someone says, the
integrity of science requires a barrier against political interference.
There you go.
Well said. Yeah.
Not going to happen, though.
Yeah.
Okay. So let's move on to our paper, which is really cool. It's a Nature article, Chromosomal Fragile Site Breakage by Epstein-Barr virus encoded EBNA1 at clustered repeats. Oh my gosh, Don Cleveland is on this paper. You guys, this is inside baseball. Don Cleveland was a thing when I was a student. He did something with gels, right? SDS
I have
Page?
no idea.
I don't think that's the same. There is something that's named after him. I looked him up in Wikipedia.
Maybe it's a different Cleveland.
I think, yeah, let me find that again.
Anyway, this is coming from UC San Diego. Yeah, all UC San Diego. We have Julia Lee, Amal Abbasi, Dong Kim, Scott Lipman, Ludmil Alexandrov, and Don W. Cleveland. You know, Don W. was the name of the original guy, and I thought he did stuff with gels. Let me look it up. Don W. Cleveland. Let me just put gels in the thing. Yeah, he did.
Yeah. So you think it is the same one?
Peptide mapping by limited proteolysis and SDS and analysis by gel electrophoresis, 1977. Yeah,
wow.
University. I remember seeing Cleveland all the time. Cleveland gels. We used to run Cleveland gels, right?
That's the year he got his PhD, 1977.
He got his PhD with Lemley, and that's where Lemley buffer comes
from. This says he worked with Mark Kirshner and graduated with a PhD in 77. So whether it was...
Yeah, okay. Lemley made up the buffer for SDS gels, because we used to call it Lemley buffer. You guys remember that too?
Absolutely.
Right, but that was way before 77. The original Lemley paper.
I'm not remembering what he did. It may be that he did the proteolysis right in the gel.
I think that's right. Right? That sounds right to me.
Mm. Anyway,
and then
we're
you
kind of
electrophorease the peptides right out of the gel.
That's
something.
right. Anyway.
Yeah. So I was thinking, this guy must be 150 years old, and he's probably younger than me.
No, he's 72. It says on Wikipedia.
Man.
Yes.
Yeah, so he's contemporaneous with. He got his PhD around when I did it. And I remember Peter Palazzi said, "Why don't we do this for RNA? Why don't we digest the RNA with like T1 in a gel slice?" Right, based on Cleveland's thing. Anyway.
Right. So here it is. He said, it says, he developed and published a peptide fingerprinting technique that was so popular it became a citation classic. And that's the development of Cleveland peptide mapping by Don W. Cleveland, JBC. This thing doesn't. That's a paper in 2006, sort of a historical look back at it. Okay.
Right.
Well, you're never too old, I guess. Good for him.
Right.
This is awesome because this is a great paper.
Yes.
Epstein-Barr virus is a herpes virus that many of us have, most of us have. We get it at a young age, and it remains with us forever. The genome remains episomal, which means apart from the chromosome. Is it linear or circular? Does anyone know?
I think it's, oh man. I think that it is linear in the virus particle and circular when it's in the latent state. Somebody correct me if I got that wrong.
Yes, I just checked. You are correct. Linear form.
Seems right.
Yes.
So, you may have infectious mononucleosis, right? One of the symptoms, but the virus remains with you forever. And in some people, it's associated with various cancers of both lymphoid cells and epithelial cells.
Like 95% of the population or greater
huge.
Has this thing. You get infected with it when you're young. Usually it doesn't do anything. It stays with you forever. And occasionally it manifests with certain symptoms, like, for example, infectious mononucleosis. That's the most common one.
Some cancer.
It's also been linked to multiple sclerosis recently, right?
Yeah, that's right.
Yeah.
We did that paper,
MS. Mm-hmm.
And Burkitt's lymphoma is one of the cancers. Nasopharyngeal carcinoma is another one.
Gastric carcinoma as well, I think.
Yep.
And so there's a viral protein called EBNA1, Epstein-Barr virus nuclear antigen 1, EBNA1, which is involved in the persistence of the genome, and it is it's one of the few proteins that's produced actually during the latent infection. And herpes simplex virus, for example, no proteins are made during latency, but here. One, and I don't know if others are made, probably EBNA2 as well and others, but EBNA1 is essential for latency. And this protein binds an 18 base pair palindrome that is repeated 20 times at the origin of replication of EBV. The origin of replication is where DNA synthesis begins. So you have this 18 base pair repeat that is binding this protein, and this is implicated in persistence of the genome. You can immortalize B cells with EBNA1. It's one of the targets of Epstein-Barr virus latency, or B cells.
I had no idea when I read that. I had never.
And so then the question is, does this protein have a role in cancer, in tumorigenesis? And that's what they're doing in this paper. And so the experiment is really, really straightforward. They take EBNA1 and they hook it up to an epitope, which they can then detect easily. It's called a flag-tagged version, but it's basically an epitope. And then they put this in cells, and the protein is made. And they use immunofluorescence, using antibodies against this tag, and they see two sites where this protein is in the nuclei. They're using a couple of different cell lines, retinal pigment epithelial cells, some tumor cell line, and HeLa cells. When they make this protein, and they're making a lot of the protein, it seems to stick to something in the nucleus. And it only works with human cells, didn't work with mouse cells.
And the fact that there's two distinct sites already makes you suspicious that it must be binding to some specific thing on a chromosome, and there being two chromosomes, you would get two spots.
Right. They never really come out and tell you that that's why there's two spots. It's kind of like a secret handshake of genetics. You just have to know that it means there's two chromosomes there.
So they figure these are uninfected cells. There's no Epstein-Barr virus in these, right? So that's Epstein-Barr virus has the 18 base pair binding site for EBNA1. So they say, it must be binding nuclear DNA. And so then they do some experiments to look at that. They delete the EBNA1 has a DNA binding domain. It's a part of the protein that binds directly to DNA. And when you delete that, no longer makes these two foci, these two punctate foci. And beautiful pictures. Really nice.
Unfortunately, this paper is not open access, but they are beautiful pictures.
So they say, this tells us there must be some of these 18 base pair sequences in the human genome. And they then look at sequences. People have previously reported over 900 binding sites for EBNA1 in human genome. And they look at that data and they find that they can see signatures of this 18 base pair palindrome in the human DNA sequence.
And it's not a perfect match.
Yeah, it's not perfect.
It's close.
And this is located on chromosome 11, in particular 11q23, one of the arms of the chromosome. And they curiously looked. So there's one cluster of these repeats, these 18 base pair repeats. Not perfect match for EBV, as Kathy said, but close enough. They looked in genomes of two individuals of Ashkenazi descent and two of Chinese descent, and they have differences in copy number. So there's polymorphisms in this sequence in the human population.
And if you've got a repeat sequence, that makes perfect sense. You can get recombinations that give you expansions and contractions of the number of sequences.
Right.
The other thing is they kind of don't get to it to the very end, but this 11q23 locus is one of 51 fragile sites that were identified almost 40 years ago in the human chromosome. And so if you look in that paper, which I did, they have it would be supplemental data now, but they have the full table of where they all are, and you go down, you get to chromosome 11, and 11q23, there it is. So the fact is that these repeat sequences that have this homology to where the EBNA1 binds in the virus, now in the human chromosome, it's there at this fragile site.
Kathy, when was that paper?
It's 1984. It's reference 25.
So I'm wondering,
paper.
How do they identify fragile sites at that in 1984?
How did they identify? So let me tell you in this paper what they did. They used some other mutagenic kinds of things. Let's see. That took it the wrong direction. I'm not able to enlarge this paper on screen. No, it's like going smaller and smaller. There, okay. They took blood from 5 males and 5 females, and then they 8 of them were normal. One had mental retardation. One had acute leukemia. We also donated blood after having taken. So it sounds like they might have sampled themselves. I don't know. After somebody took folinic acid daily for 3 days, and they studied from a 24-year-old female chimpanzee and a 34-year-old female gorilla. They used blood lymphocytes, and then, so trying to get to the answer to your question, they mapped the fragile sites. They made chromosome preparations, and then did G banding and cultured with or without caffeine and looked at breaks. So they just did chromosome spreads, evidently. They've got some pictures of spreads here.
Okay.
G-phase, banded, metaphase of human
So
cells.
this is actually just like physical mapping of
Yep.
places in chromosomes that are liable to break under certain circumstances.
Yep.
Okay.
Yep. And this is one of them, 11q23, where these 18 base pair sequences from EBV are present.
One of the things that struck me about this paper, correct me if I've got this wrong, but I got the impression that at several times in the paper, they were sort of reanalyzing, remining existing genomic data.
That's right.
Okay.
That's right.
And my sense was that in particular, in areas like this where there's a lot of repeats and that kind of stuff, the sequencing data that exists may not be perfect. And that if you really want to understand what a region like this looks like, you may have to analyze several sequences or reanalyze existing sequences, or even maybe do some resequencing yourself to get it right.
Agreed.
Back on this 1984 paper, sorry, I should have just read the abstract. They looked at these lymphocytes from 10 people, 2 great apes, when they deprived them of thymidine and looked at the incidence of breaks, which was enhanced by caffeine.
Okay.
And then they looked at 20 of the sites and found that they were correlated with breakpoints that had been related to human malignancy.
Okay.
Is that standard? I have no idea. Why would they use caffeine? Is that something that is used in, do we know? Like, I mean, is that?
Caffeine's a purine, right? I mean, that's the first thing I think of. Maybe it has some sort of
mutagenesis.
Some sort of mutagenic effect.
Yeah.
Okay.
Yeah. All right, back to the paper. They then use CRISPR to remove this 18 base pair palindrome, and it reduces the EBNA binding foci in cells. So they can say that these the EBNA binding sites are right at this cluster of 18 base pair imperfect palindromes at 11q23. So then they go into a little discussion of why any repetitive sequence, as Rich alluded to, is a challenge to genome stability, because anytime there is some kind of stress, these fragile or repetitive regions have problems being properly replicated. They make weird structures, and they're called fragile sites for that region, and they correspond to breakpoints, as Kathy says, in cancers. And one of the ideas is that when you have these fragile sites, you do things that predispose to cancer, like you get deletion of tumor suppressor genes that are keeping the cell from dividing uncontrollably and keeping them from being transformed. You can get amplification of oncogenes that cause cancers in themselves, cause cells to divide, rearrangements, and so forth. So these repeated sequences are known to be problematic. And so they looked at this more carefully. They have DNA probes that they have used, and they could see that you can get weird structures. One of them is a tiny nucleus, micronucleus. And I put a picture of this in the show notes. You can stain DNA with DAPI. It looks blue under the UV light. And you see the nucleus, and you see the tiny nuclei where if you have a fragile piece of DNA, it breaks off and it makes its own little nucleus. And they can see that in some of these cells. And they use drugs to apply stress. They use acetylcholine, for example, to induce replicative stress. And they can see that these little micronuclei are made, and that's from breaking at these fragile sites. And in fact, some of these micronuclei induced by replication stress have the 18 base pair imperfect palindrome that binds EBNA1. And so one of the ideas is that the binding of EBNA1 to this region causes the DNA to bend, and that can induce stress leading to breakage. And they have a nice picture of how that could happen. You can imagine if there are a lot of 18 base pair repeats and the protein is binding along, the protein can interact with each other and cause a curvature of the DNA, and that could impede DNA replication.
Yeah, the commentary. I don't know if we mentioned that there was a commentary also by Laurie Frappier. They have a very nice illustration of this, the figure that they have for how this could potentially be happening, the mechanism.
And so if you get a break, as I said, if it's not fixed and the piece of the chromosome remains separate, that becomes a micronucleus. And that can be propagated through multiple cycles. So they did an experiment where they turned up the amount of EBNA1 in cells using a drug to turn it on. And so without the drug, the amount of EBNA1 is low. And then when they turn up the levels of EBNA1 with this drug, you can see many more cells have broken chromosomes right at that site. So it seems to be related to the amount of protein, which is, again, consistent with this idea that it's many copies are binding and making the DNA get curved.
But it's not very much increase of the EBNA1. It's kind of like it's almost at the threshold for danger.
Yeah.
Double it, and then it's a problem.
Yep.
Yes. I think twofold is what they end up saying. You just go up twofold, and that's enough to get it over. And the idea is that in latency, you have a low amount of EBNA1. And then when a stress happens and you push the virus into the lytic phase where it's going to make particles and infect someone else, you get more EBNA1, and that is leading to the breakage. Yeah. So there's a gene near this EBNA1 binding site, so 4 million base pairs away. It's the ATM gene, ataxia telangiectasia mutated gene. It's a tumor suppressor that is often altered in cancers. Is 6 million bases away. And there's also an oncogene called the mixed lineage leukemia proto-oncogene, which is close by too. And that's rearranged in 70% of infant leukemias and 10% of adult myeloid leukemia. So these are two genes involved in cancers that are close to this breakage site. And if they induce breakage with turning up EBNA1, they can see fragments with these genes in them, and they persist over multiple cycles. So you can imagine that maybe their regulation is now deranged because of this breaking off.
Yeah, when I try and think about this kind of stuff, I rather than trying to rationalize my way all the way through steps of oncogenesis and stuff, I just think, okay, so you got genes involved in cell cycle regulation, and that if they are dysregulated in some fashion, could lead to a cancer. So it's if you stick these on a mini chromosome, you are likely dysregulating them. So cancer could be an outcome.
So this now comes the experiment where they increase the amount that we've just already discussed, but they put it in a nice way. They say, we want to see if expression of EBNA1 is kept low during latency to prevent rampant breakage of chromosome 11, right? I mean, that wouldn't be good because the genome needs to be maintained in a latently infected cell. If the cell dies, then that's evolutionarily not selected for, right? So they increase they look at the level of EBNA in latently infected cells, and it's about half what you see that's needed to induce breakage of chromosome 11. So as Kathy said, it's only a twofold difference that makes it break the chromosome. There's this other concept that I hadn't heard of before. So when 11q23 is broken by EBNA1, something called chromosome shattering occurs, and there's a word for that. It's chromothripsis. Wow. You never heard that one before.
No, that's a new one.
Mm-hmm.
You know,
I've heard of chromosome shattering, but I don't think I've heard of
chromothripsis.
I've heard of cytothripsis, cell shattering. I've heard of that before. Yeah, but not chromothripsis. No.
You know, a thrip is an insect of some kind. Yeah, which transmits some virus infections. anyway, so they say when this chromosome breaks, you're going to get shattering, making additional fragments. These could be lost. They could be religated into the chromosome. You can imagine all weird things happening. So they said this EBNA1 induced breakage is likely to cause a lot of structural variations of the two arms, the P and the Q arms of chromosome 11. All right. So what about tumors? Right? You're all asking, what about tumors? What's going on? So nasopharyngeal carcinoma has a high association with EBV among all the different cancer types that have been looked at. That's the big one caused by or associated with EBV. And so they have 78 previously generated whole genome sequencing data of these DNA from people with these tumors or from the tumor DNA. And they're all EBV positive. They did staining to confirm that. 63 out of 78 of these genomes had structural variations of chromosome 11, which is where we're breaking by EBNA1. And 32 had clustered structural rearrangements within 100 kb from one another. So all these, a good fraction of these nasopharyngeal carcinoma tumors have rearrangements right around where this breakpoint is. And I think Lori Frappier, she said in her commentary, it would be nice to have EBV negative nasopharyngeal carcinoma as a control, but they're really hard to get. And then they looked at other tumors. So what do we find in other tumors? 38 different tumor types, and they have 2,439 cancers among these 38 different tumor types, and they could find EBV in 165 of those patients, and those that had EBV had enrichment of chromosome 11 structural variations. And 100%, 18 out of 18 head and neck cancers that are EBV positive had rearrangements of chromosome 11 as well. That's pretty good.
They also found, though, that there were EBV negative tumors that also had these fragile sites could also be broken, and those that were EBV negative, which was interesting.
Right.
Not only the positive.
So they say it's possible that EBV may have come and gone, right? Hit and run.
Exactly. Yeah. Yeah.
But
Exactly.
But Lori says in her review, there's no evidence that happens.
Yeah, there's no reason why you couldn't get breakage at those sites. They are fragile sites for other reasons.
Yeah, for other reasons, right?
That's why it's called a fragile site.
Yeah.
A little caffeine, Yeah.
Oh, darn. All right, so their conclusion is our work identifies a previously unappreciated link between EBV and structural variations at chromosome 11. We propose in cells infected with latent EBV, chromosome 11 is broken by elevated levels of EBNA expressed after reactivation from latency. So that's reactivates, you get higher levels of EBNA1 and breaks chromosome 11. It's very interesting. Right?
Very cool.
Do we know? Oh, sorry. Do we know, like, EBV, like they say reactivation of EBV because it can obviously be latent for years, or in what cases, like, I don't even know. I don't know if you guys know. Would that be relevant? Because in any sort of immunosuppressed state, then there could potentially be reactivation of EBV. But is there something that's typical that we know that EBV is reactivated, or in the elderly, is it reactivated more often? Do we know? Or
it may be known. I don't know. It might be interesting to what comes to my mind is that I wonder if there are sort of I don't know what you would call them, comorbidities associated with EBV affiliated cancers that you could look at and say, I wonder if such a comorbidity could, lead to EBV reactivation that could do this. Okay. I don't know, because I was also wondering, this is a great story, and it gives us a mechanism potentially for EBV involved in oncogenesis. But in terms of, and they don't comment at all on this in the paper, in terms of, the clinic, what do you do with this information? Okay.
Exactly.
Is this, can
So-
you use this
Yeah,
to
that's a good
prevent
point.
cancers? I mean, your question about inducing EBV from latency is relevant in that regard, because that seems that's a logical trigger. So maybe what you want to do is make sure that you don't do that. Well,
exactly.
how you do
Considering what
It's tough.
99% of people are infected with EBV,
Yeah.
then
Yeah,
basically everyone.
no, you're never going to get rid of the EBV.
Yeah.
So you could CRISPR it out, but I don't know that you're going to do that because of the off-target effects and so forth. So, but that would be the solution, right? But you get infected again, right?
Yeah,
because
and
it's everywhere.
it's just like you could theoretically get rid of the HIV provirus in people infected with HIV, but you're not going to do that.
No, there are too many off-target effects.
Right.
That's the problem. So I found an article in Journal of Clinical Pathology on what reactivates EBV. EBV reactivates under conditions of psychological stress with consequent weakening of cellular immunity. It occurs in a subset of individuals with each of a variety of cancers, autoimmune diseases, autoimmune-like disease, chronic fatigue syndrome, and other circumstances such as being an inpatient in an intensive care unit.
So that read that again. That can't be psychological stress. Physiological stress?
Psychological stress.
Psychological stress.
Yeah.
So stay cool.
Easier said than done. But if you go to an ICU, you have a high likelihood of having your EBV reactivated.
That makes perfect sense to me.
It could be the stress, the psychological
Just
stress,
all kinds of stress.
or physiological. Yeah, whatever's wrong with you.
But I mean, prolonged stress would suppress your immune system. So if you have like this chronic stress that's suppressing your immune system, then it could reactivate.
So you could use an antiviral to inhibit lytic replication, right? But you don't know when you're reactivating. For herpes, some people get a cold sore, and so that could be a sign. But I don't know that you would know with EBV that you're reactivated. So it's a tough one. I'm not sure that this has any actionable information other than it being really interesting and explaining, what might be going on.
I'll bet you somewhere down the road, outside of our limited imaginations, it is actionable.
Maybe.
You never know.
So. Yeah, I'm sure it will. I just can't think of anything to do with it at this time, but some smart person will.
Some smart person will. I have to comment, too, that this paper has 4 figures and 14 supplementary figures.
Yep.
But it only has 6 authors. It has 6, not 30 or 40 or 50.
Stuff.
I was going to comment on that too, considering it's a Nature paper and there's only 6 authors.
Yeah.
It's a lot of data too. So,
it's beautifully done.
And yet, did we say the first author? Did we mention Julia Suzu Lee? Just to
I did. Read them all.
Okay, perfect.
But that's good. We can give her another shout out because it's very cool. All from UC San Diego.
Yeah.
Where, there's no stress because it's nice all the time.
I was in graduate school there. I would say there was some stress.
Isn't it expensive to live there? That's stressful.
There is that too. But, as graduate students, we had some university subsidized housing. So that's made it affordable.
But Kathy,
I can experience stress anywhere.
I would like to see a map of the U.S. state per state at the rates of EBV-induced cancers. I bet they're lower in California. I'm just kidding. But if you get stressed at UCSD, you go outside and it's beautiful out. You sit outside for a while, get some sun, right? Maybe not.
Maybe.
72, yes.
I went off on a tangent because we talked about chromothripsis. And it's a neologism coined by the scientists at the Wellcome Trust, derives from chromosome and thripsis. Thripsis is the Greek word for breaking something into small pieces.
Ah,
So then I looked that up in Wiktionary, and they even give it in Greek letters, and it comes from thlibo, which means to press, and the suffix for cis. Okay. Then I looked up thrips etymology, and that's a different set of Greek letters, and it means woodworm. It's a direct transliteration from woodworm, and then I put in the picture of the woodworms for you.
yeah,
So-
very good.
So I think thrips and chromothripsis are not related.
Sounds like to me, yeah.
All right, let's go back
Nevertheless,
to
the woodworms would make a nice episode image.
let's go
Right.
back to actionable. First of all, I went back and looked at the connection between EBV and MS, and there's an article
Mhm.
NIH says at the end, it's a big step because it suggests most MS could be prevented by stopping EBV infection, right? So people are thinking about that.
Right.
So if you stop that, you can stop cancers also, right?
But everyone's going to be infected with EBV. That's impossible to stop infection.
You can't stop infection. You could, yeah, if you allow, so let's say you had a vaccine and you got vaccinated in your early years, it would still allow infection.
Hmm.
So probably that's not enough because remember the MS is an autoimmune thing. It's an antibody against one of the viral proteins. I'm trying to remember if it's EBNA1. Do you remember if it was EBNA1 that
I think it is because when we did the paper, I remember hearing EBNA1 and I was like, why have I heard this before? And I'm going to double check.
So I'm thinking, well, could we make a vaccine against EBNA1? No, it's going to cause autoimmunity in a fraction of people, right? So that's not good. I mean, so some of the antibodies against EBNA1 cross-react with a neural protein, and that's the idea, the etiology of MS.
It
So
is
you-
EBNA1.
So making a vaccine that targets EBNA1, and-
Well, could you make it to like a very specific epitope of
Maybe.
EBNA1 and not get that cross-reactivity?
maybe. And the antibody, of course, would have to get into cells also, right? Because the EBNA in the nucleus is what's doing this problem, this chromosome
Oh.
breakage. So that's a big ask, I think. You can make a small molecule that blocked EBNA1 binding to DNA, probably, right? But I don't know if you want to give people small molecules their whole life, right?
Right.
And I'm just thinking out loud.
Yeah.
Right.
Yeah.
Yeah.
We're not going to live long enough for this one.
Probably not.
Well, us,
Angela, maybe.
Angela will.
Maybe.
She's got a head start on us.
Yeah, okay.
Do we know how stable EBV is?
What is
Like as
what
a
do you-
virus, is there a lot of recombination? Like I'm not sure. I actually know very little about Epstein-Barr virus in general.
I'm sure
Where are you
we can
going
You
on
buy
mean
it. What do you-
genomically stable?
Genomically stable, sorry, is what I meant. Like it is what is it? It's a herpes virus, right? Yeah.
Yep.
So then yes, more so than okay. So more
Yeah,
or
it
less.
does. It does
stable.
recombination. Yeah, there are articles on it. It does it all the time. Yep.
I was just thinking of like how I'm not sure if the Epstein-Barr virus in me versus you, how genetically distinct they are, and how obviously like HIV is very
I see. So
heterogeneous even within one
phylogenetically stable is
population.
what you're interested in. I think
Hmm.
much more stable than RNA viruses, right? But I don't know. Boy, man, I feel like we've addressed this before. Done populations of herpes viruses, but maybe not.
So studies in herpes viruses like simplex support the idea that recombination are associated with better capabilities to evade host immune surveillance. The role of recombination in altering risk of EBV-associated malignancies is unclear given the lack of properly controlled investigation involving cases and controls. So they do recombine. And this study, which I'm looking at, profiling genome-wide recombination in EBV, reveals type-specific patterns and associations with EBV, with endemic Burkitt lymphoma. Sorry. This is done in Western Kenya. So, yeah. Someone asked me...
I was just thinking about how easy it would be to make a vaccine if I'm not sure what sort of efficacy the vaccine would have, apart from the actual delivery method. I mean...
Yeah, I think the problem is unless the vaccine totally blocks infection, you're still going to get some establishment of latency, and that's it. Then those cells are going to divide, and you're going to get the EBNA1 and the fragile chromosome
MS.
stuff, you know?
We have a couple of different herpes virus vaccines, right? I guess.
Which vaccine?
I'm thinking of the chickenpox vaccine.
Okay.
Right. There's a live attenuated chickenpox vaccine, and there's the shingles vaccine, which is a subunit vaccine. Now, of course, the shingles vaccine, it's too late by then because shingles is reactivated. Chickenpox. It makes me wonder whether the attenuated chickenpox vaccine, it establishes latency itself.
Yeah,
and I don't know that it prevents infection or establishment of some level of latency by the chickenpox itself. I suppose if you had, I don't know, if you had an EBV vaccine, no, it's not. It's not going to. It's not going to prevent latency.
The attenuated varicella vaccine does establish latency, and it can reactivate at a very low level, right? Less than wild type. So
the vaccine itself, and what I don't know, I would imagine that it doesn't even prevent some level of infection by varicella, the original virus, and some level of latency itself. So that's probably known.
Yeah.
Somebody will write in and help us out with that one. Dave Bloom, you listening?
But if you had, my understanding is that they're looking into giving kids Shingrix for chickenpox, right?
Yeah, that makes sense to me.
To eliminate, but if it doesn't prevent infection, it probably doesn't. You're still going to get some latency if you get wild type virus, right? It'll probably knock it down.
Yeah,
right?
Maybe. But I don't know if you establish any latency at all over time. I wonder, in terms of the total amount of latency in your body.
Yeah. Do we know, the amount of latency, if that, has anyone ever studied?
I don't know. I mean, it
how
seems to
that
me
could
it
affect
could be that the way the vaccines work is just give you enough antibody or whatever so that when you reactivate from whatever pool of latency you have, the chances that you're going to become symptomatic are reduced.
And it's different from EBV, where the target cell is a B cell, and those are going to divide. So even if you infected a few and they got latent infection, they would divide. But for VZV, it's neurons, which are not going to divide.
Good point.
Right? So
good point. Maybe you could tamp that down. Good point.
Virology is really interesting. We should do a podcast on it. I mean, it's just great. I love it. Okay, let's do some. Oh, look at the thrips.
Yeah, I was just going to say I liked the final paragraph of this paper where they kind of summarized everything. What was a little frustrating to me is that in the long text section, they didn't have the headings that were kind of telling you this is showing you this, and they didn't have sort of summary sentences at the end of each paragraph. So I had to work harder on this one, but.
Yeah. I thought the commentary by Frappier was really good, though.
Yeah.
Really, really good.
Really good.
Epstein-Barr virus is a common herpes virus associated with several types of cancer. How exactly the virus affects the development of these cancers is unclear. But tumor cells are known to be derived from infected cells that express a small subset of EBV proteins. In fact, the only viral protein expressed in all types of EBV-related cancer is EBNA1. It's good. I like it.
Yeah.
Okay. But thrips, I think, spread Schmallenberg virus.
Oh.
Remember?
Could be.
He's looking it up, folks.
I'm looking it up because... Yes.
Oh, yeah.
God, Vincent.
You know why I know that? Because you remember Richard Elliott? Unfortunately, he passed a number of years ago. I once asked him, I think it was on a TWiV. I said, what is the vector of Schmallenberg? And he said, thrips. And I said, the way he said it was just memorable. Thrips. And actually, at the time, I'd never heard of a thrip.
Aren't they also
Okay.
called midges? Or is that the same thing? Isn't it also like a midge? People call them colloquially, I think.
I
Because
think so.
thrips I had never heard of. But
Thrips are, I don't think it's a midge. Oh,
Yeah.
midge insect.
What's the common name for
small
thrips?
fly?
Thunderflies, thunderbugs, stormflies, thunderblights, stormbugs, corn fleas, corn flies, corn lice, freckle bugs, harvest bugs, and physopods.
all right, we're way out of our lane here, folks.
Oh yeah. Oh
but,
yeah.
but not midges.
Not
Sorry.
midge. Okay.
All right, now
Schmallenberg virus. Wow.
Schmallenberg. You remember
I
that?
haven't heard. Yeah, I just pictured a sheep when you said it. That's all I could think of. I was like, I learned that once. That was in sheep.
Yeah.
Orthobunya virus.
It's a new, it was an emerging virus that had never done that before, right? And it spread throughout Europe. Started in Schmallenberg, Germany, right? Spread, I think, even to the UK, right? If I remember. But yeah, causing spontaneous abortions in sheep.
Exactly.
Yep. Okay, let's do some picks. Angela, what do you have this week?
Okay, I get to do the pick first. Wow.
You're first on the list here. Why not?
So I actually chose a pick of someone that recently. So it's kind of a pick of a podcast / effort of two scientists, actually, Itai Yanai and Martin Lurcher. So Itai Yanai is from NYU. He's a professor at NYU, and Martin Lurcher is from the University of Dusseldorf. And so they have this thing called Night Science. It's a podcast. They have workshops, editorials, and he came actually to give a talk at McGill last week. And he gave a talk. So he gave a talk on his work, but then he also gave a talk on the scientific process. And he defined this. Well, so it wasn't him specifically. It was actually, I think it was François Jacob that actually defined this day science versus night science. I don't know if you guys have ever heard this used, but day science, you can actually get all of the slides on the website. So you can go to nightscience.org and you can actually receive. So they have these workshops, but all of the slides are available online. And basically they have researchers on their podcast and they talk about the scientific process, about hypothesis generation versus hypothesis testing, which would be day science. So, but basically what they say is that during the day, you go and you do all of these experiments, you test all of these hypotheses, but the fun stuff, which is what they say, happens at night, which is not necessarily at night, but they call it night science. And it's the generation of these hypotheses, and it's more metaphorical as opposed to precise, like when you're doing things during the day. At night, you can let your mind wander. You have questions and contradictions, and your mind is going all over the place. And the actual citation from the book, François Jacob. So he was actually the discoverer of the principles of gene regulation. I don't know if you guys have heard of him. He died in, of course we have. I mean, for those that haven't, listening to the podcast, he died in 2013. So the citation from his book, the Statue Within is the name of his book. It was an autobiography. Says, night science wanders blind. It hesitates, stumbles, recoils, sweats, wakes with a start, doubting everything. It is forever trying to find itself, question itself, pulls itself back together. Night science is a sort of workshop of the possible where what will become the building material of science is worked out. So,
excellent.
Literally, Itai and I gave this talk. He spoke about this idea of night science. He has this podcast where he talks with researchers about their scientific process, creativity. And it was one of the most inspirational talks I have ever listened to. And I just would like everybody, if you have time, to go to their website, to just even look through the slides. He's just, the things that they're doing are amazing, him and Martin Lurcher. And I think that it's wonderful to think about that. And it kind of gets you more excited about science, listening to other people talk about their creative process and how they generate ideas and how they bounce ideas off one another. And yeah, I thought it was just a beautiful thing.
A cool little click inside this is this, I don't know, one of their articles or talks. What is the question?
Hmm.
It
Mhm.
starts off, the greatest single misunderstanding about science by the public is that scientists solve problems. In reality, scientists are primarily concerned with creating them. That is, creating problems. I like that.
Mhm.
Yeah. There's a lot of things
Sure.
like that. It's wonderful. And the slides are all free. They're on, like I said, they're on nightscience.org. So please go and check them out in the section of
Cool.
creative, the creative scientific process. So it kind of gets you
Excellent.
So, of course, I had to go back and figure out what year did Francois Jacob get the Nobel Prize? 1965 in
65.
physiology or medicine with
Yeah.
André Lavoie and Jacques Monod. So,
I started reading The Statue Within a couple of months ago, and
Oh, yeah?
So Ann Skalka, textbook co-author, gave it to me a while ago, and she says fabulous. So I started reading it, and I remember that quote that you just read. It's in very early on in the book, and it's all about his philosophy. There's very little description of what he did. Actually, it's about. And I'm thinking I could never write something like that. I don't have enough of a philosophy just to be able to do that, right? But I do think that you mentioned something, talking with other people to get you excited about science. That's what we do here, right?
Of course.
On this program. And we try and not just get each other excited, but also our listeners excited as well. So you don't want to keep it to yourself, right?
Well, you also don't want to, you got to be conscious of your do to think ratio, right? If you can be too busy to actually have that time to let your mind just drift and let the stuff bubble up.
Where do you do that? Can you just sit in the lab and do that, or do you need to go somewhere else, you think?
I think you need to go to Starbucks or something. Go home. Just go
somewhere
home.
else. Just go out.
Sit in the garden. Go
you
look
could
at it.
Sit outside.
Go talk to a tree.
I agree.
We were talking about it, how in grad school, at least as a PhD student, that a lot of us afterwards when we were talking felt that it's a lot more difficult to do more night science in grad school because you feel like you always have experiments to do. You're always in the lab. You always that maybe and also there's a certain level of, in your undergraduate degree, you kind of develop the tools. And then in the beginning of your graduate studies, you kind of build a frame where you can then generate ideas, because it's also, you can't hypothesis generate if you don't even have the bones, right? So, but I think that the idea of that is just beautiful. Or when we have moments like me with my supervisor, when we have these moments of night science and hypothesis generation, it's so exciting, right? You start throwing ideas out there and then maybe half of them don't work, or then you remember it wouldn't make sense because of something else or whatever, but it's still just fun to do. And I think it's really important to, like you said, Rich, make time for that. Even if it is just staring at the sky or in the shower. That's what Itai said during his talk. He's in the shower. We all think of these things.
Cool. That's very cool. Yeah. I like that. Kathy, what do you have for us?
I picked something that's a 17-minute listen from the radio program 1A. It's about Kermit, Jim Henson, and in particular about Rainbow Connection. And I happened to catch it. It's a replay. It's at least a year old, and they were just replaying it. And I caught it, and then I went back, and I think I've listened to it 4 times now. And there's also a 3.5 minute video of Kermit singing the Rainbow Connection in the pond. And they talk a lot about various things about Jim Henson, but about how they filmed this, and he was in this 55-gallon drum, and it sounds life-threatening the way they had to do it, because he was kind of breathing through a snorkel-like thing. Oh my goodness. But I think if you just want an uplifting moment, you could play this short video of Rainbow Connection, and you could also listen to this radio story. It's really cool.
I listened to the whole thing. Kathy, I listened to the song. I listened to the video clip that you got here about Henson and et cetera. And I actually looked up the lyrics because I'd never paid much attention before.
Yeah.
Right.
And it's delightful.
Right.
It's just absolutely. Yeah. I also looked to see if there was a barbershop arrangement that we could use.
Yeah, interesting. That would be cool.
Yeah, I found a couple. I wasn't too impressed. But somebody You know, one of the I know there's is it Simon who's the barber shopper out there in New York?
Oh, the one who does those acapella science? Is that what you're thinking
no,
of?
there's
Yeah.
yeah.
Oh no,
At any
something
rate,
different.
doesn't
Okay.
matter.
Yeah.
So yeah, there needs
Yeah.
to be a good barbershop arrangement, a rainbow connection.
Did it bring a little tear to your eye
Oh
at
yeah,
any
it's
point?
very, very good.
Poignant.
Just
Okay.
really,
Good.
and Kermit, it's perfect for Kermit. You know,
Yes.
he's
Yeah.
really, really good.
Yeah.
Cool. Neat. Rich, what do you have for us?
I have a what the New York Times calls a video opinion titled Alexander Skarsgård explains the answer to everything. Parenthesis, it involves doing some math. Alexander Skarsgård is sort of peripheral to this. He's an actor who narrates this video. Okay. The key player in this is a guy named Partha Dasgupta, who's an economist at Cambridge University who was charged, he had I guess a government contract to come up with an economic, a better economic way to evaluate the success of an economy in our current circumstances, in particular environmental circumstances. And he produced a 600-page report that has been lauded up and down globally. The United Nations awarded him the title of Champion of the Earth for his work, and King Charles III has made him a Knight of the Grand Cross, etc. So this is an impactful a piece of work, but of course extraordinarily complex. And this video explains it, along with interviews with the author that is really amusing. The video is so well done because it's really engaging, really entertaining, and explains it. And I just want to very briefly describe what this is about, using in part the most powerful metaphor that the author himself uses. And the deal is that most economies, most nations express the success of their economy by talking about the gross domestic product. But that doesn't take into account the cost to the environment to generate that GDP. And his point is that in order to really evaluate the success of an economy in the current context where it's clear that globally we're trashing the planet, you have to take into account the damage that you're doing in the process of creating that GDP. And his paper is all about a mathematical model to do that computation and figure out really what the success is and what variables you have to tweak to make it genuinely successful in a global context. And the metaphor that he uses that I really like is a soccer game, football for you non-Americans, which is, he says, if you're monitoring your team and you keep track only of the goals you score and not the goals that are scored against you, you have a false impression of what's going on. So his mathematical model scores both the goals and the goals against you. But I recommend the video. It's delightful. And I have thought about this kind of stuff in my own way for a long time. It just drives me nuts when people are touting the growth of their economy. And I'm saying to myself, wait a minute, there's got to be another metric, because we can't just grow forever because we're using everything up. All right. There has to be another way to look at this, that's where we measure our success in terms of sustainability, not growth.
Yeah.
Okay.
Aren't you tempted to read the book?
What, his report? No.
Yeah. You're not?
600 pages.
600 pages of, I'm sure, very complex stuff.
All right. Well, it sounds interesting.
The video will do it for me.
You can put it into ChatGPT and get a summary.
Ah, good. Okay.
Copy paste.
You know, I was fooling around with ChatGPT, and you can get different answers depending on how you pose the question.
Oh, yeah.
I wanted to know about whether B cells need to see antigen in MHC, right? And the answer is they don't. They can see it. They can see antigen because it's going to bind the B cell, the antibody with that. They don't need MHC to do that, right? But gave me two different answers. And then this is the funny part. I asked him or her, whatever, it. Who was Amy Rosenfeld? And said, oh, this is medical officer at the FDA who has published these two, and it gave two papers that don't even exist.
Oh,
The names of two papers that don't even exist. And then it said,
interesting.
She's been at the FDA since 2015 and was previously at Harvard and NIH. Which,
wow.
Where are you getting this from?
But I think that with people, I think there's privacy, like firewalls or some sort of, I'm not sure how it works, but anytime I've asked anything about any person, it always just makes it up. The fact that it even said the FDA is surprising.
Well,
because
if you search me, you get all the right stuff as far as I can tell.
But I think it's because you're a public figure, like you're an actual public figure, recognized public figure, as opposed to someone that's like
maybe.
I mean, you have a Wikipedia page probably, right? And
so what was interesting, if you just say, who is Amy Rosenfeld? They said, I don't have any idea who she is. And then I said, who is Amy Rosenfeld who trained with Vincent Racaniello? And then it spewed out a lot of stuff, which most of it was wrong, you know. So I don't know the algorithm, how it works. Someone said, you know, it looks for a word and then it looks for another word that matches and on and on and on. It just keeps doing that for billions of times. So maybe if you get misled early on, it's very interesting. Very interesting. Okay.
Something useful. Oh, sorry. About ChatGPT. One more thing very useful is that, you know how sometimes you find a YouTube video, at least I consume a lot of YouTube. And sometimes it's like an hour and you don't have time to watch the whole thing, and watching it on 2x speed is just unnecessary sometimes. So you can actually, you know how you can take the transcript? So you can actually copy paste the transcript from the video into ChatGPT and just ask for a summary. And it'll give you like a nice, like 1 page or however long you want the summary. And that's actually very good.
You can give it the whole transcript and say, summarize this.
So you can do it with a TWiV video, like with a podcast. If somebody didn't want to watch for 2 hours, they could literally put in our transcript into ChatGPT and get like the main points of our conversation.
I don't know. A transcript of TWiV? What are the, you know, that's going to confuse the hell out of ChatGPT.
I'm going to test it. I'm going to test it.
Try it.
Well, no, but it just takes the transcript, right? It literally it'll give you like the best summary. I'm going to try it with this episode.
Okay.
So, good. Rich, you know that YouTube generates a transcript automatically, right?
I do now.
Yeah. I didn't know that either.
Oh, sorry.
Where do you find it or get it?
It's right there on the page. What is it? Below, I think, you say show transcript and it will show, and you can copy the whole thing and paste it somewhere else. Yeah.
Exactly.
But it doesn't have any of you don't know who's talking. It's just a flow of.
It's just information. Exactly. It's just the words.
Yeah.
But.
Well, speaking of 2-hour podcasts, so I started a new podcast, which is going to be no more than 10 or 12 minutes per episode. And last week, my pick was Paul Offit's post on Substack called something about, nasal vaccines, hope or hype. His Substack is called Beyond the Noise. So I was waiting for the train one morning. I said, why don't I ask him to do a video summary of that? That would be great. And so I emailed. He said, yeah, that'd be fun. So Tuesday we recorded it. I edited it. I put it up. And there it is, episode 1 of Beyond the Noise with Paul Offit.
Cool.
I'm hoping he writes them every 2 weeks. I'm hoping to do it every time he writes it. And people seem to like it. So check it out. It's on YouTube only. It's going to just be a little video. And I've always wanted to make short stuff. So we'll see if it sticks. Unfortunately,
the whole series is going to be called Beyond the Noise.
Yeah, and then it'll have a sub name of his blog post. So this one is, COVID nasal vaccines, hope or hype.
Right.
So only with Paul, right? Only with Paul Offit for now.
Yeah, that's for now. And I can make others with other people, but I thought this would be. I mean, he said to me, Vincent, after you picked my Substack last week, I got a huge increase in subscriptions, I said, yeah, it's a TWiV bump. We've known that for many years. So he loved it. And then after the video went out, he tweeted it and he got a lot of positive comments. And so he's very excited. So I think he's got a nice way with words, right? He's very clear and
definitely.
so forth. So I got very excited to do that, and wasn't, I was at a meeting in the afternoon at an Infectious Disease Society of New York meeting, and I was sitting there editing it while I was listening to the talks because I wanted to get it up to post it.
The Virulent Vaccine Wars documentary has Paul Offit in it. So you might have to give up on your dancing. Probably not, though.
I can see it somewhere else, right?
I think you have to find somebody who's going to pay to rent it. So at the moment.
So ASV paid to rent it?
Yes.
Oh, okay. Well, I could do a showing here in New York. I could rent it and show it in a theater. I know a theater. I bet people would come. And then we could have a panel afterwards and talk about it.
Right.
That would be cool, like a 92nd Street Y kind of thing. That's the sort of thing they would do. So much can do. Oh, boy. This is fun. All right, that's TWiV-1007. Show notes, microbe.tv/TWiV. Questions and comments, twiv@microbe.tv. If you enjoy our work, we would love your support so we can do more, like make a Paul Offit series. We can do other things as well. Microbe.tv/contribute. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Angela Mingarelli is at McGill University. That's in Montreal, Canada. Thank you, Angela.
Thank you. Have a wonderful weekend, guys.
How do you say Montreal?
Montréal.
You say Montreal. Yeah, that's the real
Montreal.
way. Us Americans, we say Montreal.
You can say Montreal. Montreal.
When I say Montreal, the people say you're being pretentious,
Or just saying it in French.
Yeah. Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.